Welcome everybody to Evolution Prezi number 4, Measuring Evolution, aka Enter Math. These two equations you are going to use for the rest of this chapter. Big questions: how is variation generated and maintained in a population? How do we know evolution is happening in populations? What aspects of a population contribute to evolution? How can evolution be qualitatively and quantitatively measured? How does measuring evolution help us understand how populations are evolving? These big questions can be answered in this Prezi. They also can be answered from your textbook as well as the video resources. Please do make sure that you're able to understand these questions and that your notes answer them. So first and foremost, I want to take a look at how this Prezi is organized. So we'll discuss how populations evolve in general. We'll discuss variation. We're going to review some required terminology. To understand the information in this presentation will require you to think back to your freshman year of biology. So this is more of a review notes, if you will. Over here, this is the meat of the information. This is the 5 sources of evolution. We've discussed many of these things already, so it's kind of putting it together. And finally, this is the application of Hardy-Weinberg equilibrium. So this is actually how we measure evolution. And here you're going to have some practice problems to do. So you'll be pausing this video so that you can answer those problems and bring those answers to class with you next time I see you. The last part of this presentation discusses something called heterozygote advantage, which is a real-world application about how being a heterozygote for something could be beneficial to you. So let's start with populations evolving. So as a reminder, individuals are selected via natural selection. And so if you have differential survival, that means that you're going to have differential reproductive success. It's up to you whether you want to write this down again or just highlight it in your older notes. The second piece of information that's very important, that again is a review, is that populations evolve. So individuals are selected, but populations evolve. And so it's the genetic makeup of a population that changes over time. As the fitness increases, the favorable traits that give you a greater fitness or give an organism a greater fitness become more common. Variation is the raw material for natural selection. There have to be differences within a population for natural selection to be able to work. Some individuals must be more fit than others. So where does this variation come from? So ultimately, all new variation comes from mutation, which are random changes to the DNA. The why we would have random changes to DNA is something that we'll discuss a little bit later on, but know that there are mistakes that are accidentally made in the copying process of DNA replication. And then variation also comes from sex. So mixing of genes or recombination results in new arrangements in every offspring, which is why if you have siblings, your siblings and you are different or vary in the traits that you have and the genes that you have. So offspring are going to inherit traits from parents. So some required terminology that, again, you've heard before, but we're going to go over again today, include gene. So genes can determine a trait. For example, the eye color gene or genes. An allele is a variant of a gene. So for example, brown eyes or blue eyes. You've also heard of the term dominant and recessive that we'll be discussing a little bit later on. Now, it's important to note that a gene is located on a chromosome, or multiple genes would be located on multiple chromosomes sometimes. So all sexually reproducing organisms have 2 alleles for any trait. You have varieties include a dominant allele. We typically will write this as a capital letter. So it's an allele that will show a trait regardless of the other allele. A recessive allele is another type of allele. That's an allele that will only show a trait if both alleles are recessive. Typically, we write dominant alleles as a capital letter, and we write recessive alleles as a lowercase. So again, dominant alleles are represented by capital letters, recessive alleles are represented by lowercase letters. Since all sexually reproducing organisms have two alleles for any particular gene or trait, there are three different options. So we've got an organism could be heterozygous, or an organism could be homozygous and have two copies of the dominant allele, or an individual could be homozygous and have two copies of the recessive allele. So an organism that's homozygous is any individual who has two copies of the same allele. This could be, again, homozygous dominant or homozygous recessive. If you're heterozygous, that's an individual who has one copy of a dominant allele and one copy of a recessive allele. And you are typically going to show the dominant trait. Some other terminology that you should be aware of is population. And so a population is a localized group of interbreeding individuals. A gene pool is something you're going to hear me refer to a lot, and it can be helpful to visualize, or maybe you will want to draw in your notes, a gene pool with a bunch of different alleles in it, like we did in class the other day. So this is a collection of alleles in the population. Do remember the difference between alleles and genes. So the alleles are going to be represented by a capital or lowercase letter. There are five sources of evolution, as we discussed previously. The first is mutation, the second is gene flow, the third is sexual selection, the fourth is genetic drift, and finally the fifth is natural selection. So mutation creates variation. New mutations are constantly appearing. Mutation changes the DNA sequence, which will change the amino acid sequence, which will change the protein structure and function, which can change traits and potentially change fitness. That may or may not happen, that last piece. Gene flow is the movement of individuals and alleles in and out of populations. So seed and pollen distribution by wind and insects, the migration of animals. This is going to reduce differences between populations, or in other words, decrease genetic variations among neighboring populations. Gene flow in human populations is increasing today thanks to modern travel technology. Here you have an example of some gene flow that you are probably familiar with. All right, number 3, sexual selection is definitely something that can cause evolution to happen. Genetic drift, remember that this is the effect of chance events, so that includes founder effect as well as bottlenecks. Loss of alleles from a gene pool results in reduced variation, and it also will reduce the adaptability of that population in that particular area. And so here, what we can see, this is really interesting to look at. So if you look here, the C is the gene, and this is going to be like flower color, okay? And so here, the flower color for R stands for red. The flower color allele is R. So in the gene, the C gene for color, this red flower has 2 red alleles. Looking at this white flower, in the same gene, the C gene, the color gene, we have W and W. That stands for 2 white alleles, thus producing white protein. Down here, this is pretty interesting. This is not something that we discussed in freshman biology, but we will be elaborating more now in AP biology. This is something that both the red and the white kind of seem to mix together, if you will. And so this is a heterozygote. It has both a red allele and a white allele. So here we don't have complete dominance like we do typically when we think of whenever you have a recessive and a dominant allele, you would have, only the red or only the white showing. But in this case, since we have a pink combination, flowers are an example of something that we call incomplete dominance because the red is not completely dominant to the white, so it's incomplete dominance. So if you look here in generation 1, you can actually count how often you see one allele over the other. And so if you count them all up, one red, 2 red, 3 red, 4 red, 5 red, 6, 7, 8, 9, 10, 11, 12, 13, 14. So there are 14 red out of how many total alleles? Let's see. 2, 4, 6, 8, 10, 12, 14, 16, 18, 20. So 14 out of 20, that's going to be 7 out of 10 or 70%. So in frequency, that's going to be less than 1. So you can represent a percent in decimal form by saying 0.7. And so we're going to represent that as the frequency of 0.7. So 70% of the alleles in generation 1 are that capital R, that red allele. So 5 plants are going to leave offspring. And if you look, generation number 2, we see that this red allele is now down to 50%, whereas the white allele went from 30% to 50%. 2 plants are leaving offspring, and now in generation 3, we see one, meaning like this generation here actually only has red alleles and it does not have any white alleles. And this is due to genetic drift. So this population is so small that it's very easy to drift in one direction or another. In this case, it drifted toward the red alleles, and this was not due to them being better suited to the environment. It was literally just chance of reproduction. Natural selection, of course, is the differential survival and reproduction due to changing environmental conditions. So different combinations of alleles that provide fitness increase in the population. And this is adaptive evolutionary change, meaning organisms that are selected for by natural selection, we will see their populations evolve to be better suited to their environment. Okay, so what conditions would cause allele frequencies to not change? So these are all things that can cause evolution, thus changing allelic frequencies over time, like we saw in this example of our flowers, the genetic frequencies changing over time. If those conditions cause allele frequencies to not change, to change, then what conditions would cause allele frequencies to stay the same? Okay, so in walks Hardy-Weinberg equilibrium. Okay, so this is going to describe a hypothetical, non-evolving population that preserves allele frequencies, meaning all of those things that you wrote up above that can change allelic frequencies. Hardy-Weinberg equilibrium, the only way that this math works is if you say that none of those things are at play. So what we do, this is not a real population, but what it serves as, or what we use it for, is a model for comparison, a null hypothesis. We'll discuss more about what that is. This is important, this term that you know, a null hypothesis. So it's going to serve as that model of comparison. Natural populations, no, are never in Hardy-Weinberg equilibrium, but it is, again, a useful model to measure, to compare if forces are actually indeed working on a population. So this is important. The Hardy-Weinberg theorem assumes that there are only 2 alleles. So you've got both the dominant allele and the recessive allele. And so we represent the frequency of the dominant allele as the letter P. We represent the frequency of the recessive allele as the lowercase letter q. Now, frequencies have to add up to 1 or 100%. So we know that P plus Q equals 1. Now, pause the video here if that's not making sense, and I will do my best to explain it again. So we know that there are only 2 allele options, right? So you have a dominant allele and you have a recessive allele. So let's say the dominant allele makes up 30% of the population. Well, that must mean that of the gene pool, excuse me, that must mean then that 70% must then be the recessive allele in that gene pool, because you have to have 100% of your alleles accounted for in that gene pool. So the frequency of the homozygous dominant is going to be represented by, this is actually not coming out as accurate, so I'm glad that we're discussing this. So this is p squared is the frequency of homozygous dominant. The frequency of homozygous recessive individuals, that would be q squared. And then the frequency of heterozygotes is represented by 2pq. So frequencies of all individuals must also add up to 100%. So there are two equations that were written at the beginning of this presentation. So we've got the frequency of the dominant allele, the frequency of the recessive allele must add up or be equal to 1, as well as individuals that are homozygous dominant, individuals that are heterozygous, and individuals that are homozygous recessive. Those must also equal 1 or make up 100% of the population. Please make sure that you have these formulas written down, and in class I will write on top of these to help you remember what each of. So as I mentioned before, I'm not going to be solving the Hardy-Weinberg problems with you in this format because it is not the most how to do these problems. However, I do expect that you attempt to do these problems in the Prezi. There's not enough information potentially in this Prezi for you to be able to answer those questions. So my advice is read the textbook, this section, or you need to watch the Bozeman Science video on how to solve Hardy-Weinberg problems, because we are only spending one day in class reviewing this material. So it's imperative that you spend the extra time now digging deeper into the content. The one thing I do want to touch base on is your advantage. And so here's an application of a Hardy-Weinberg principle, sickle cell anemia. So due to a mutation in a gene coding for hemoglobin, which is an oxygen-carrying blood protein, recessive allele, coded for HS, makes a defective protein. The dominant allele, which most people in the world have 2 copies of, HB, makes the normal protein. And so it is a recessive disease, meaning individuals must have 2 of these to have sickle cell anemia. Some of the symptoms include low oxygen levels, which causes the red blood cells to sickle. That's what they look like here. Breakdown of those red blood cells, clogging in the small blood vessels. This is a very painful disease because unlike the normal red blood cells, sickle cells do get stuck in places, especially in joints, and can cause a lot of pain. And so ultimately that will result in damage to organs, and it is often lethal in childhood because your body needs oxygen. And so this is not an effective way to transport that oxygen. So it turns out that sickle cell frequency is different depending on where you live. So there's a high frequency of heterozygotes, and oftentimes in communities, people refer to having the trait, otherwise known as sickle cell trait. They just say that rather than being, saying like somebody is heterozygous. In African populations, so 1 in 5 Central Africans actually are heterozygous for sickle cell anemia. And this is unusual for allele with severe detrimental effects. So you would think that natural selection really decrease the amount of that particular allele, of the recessive allele, since it is so harmful. However, 1 in 100 people have the sickle cell anemia, and unfortunately they usually pass away before reproductive age. The cool thing is that in tropical Africa, malaria is common, and so heterozygotes actually have an advantage. They confer resistance to malaria because the parasite is not able to live in those sickled cells, and so they have enough oxygen to basically transport around because they have enough normal red blood cells. But anyway, that means that if you're heterozygous, like there's some resistance to malaria. If you are homozygous dominant, meaning you contain both of the quote-unquote normal red blood cells, or the the normal hemoglobin, proteins that are in those red blood cells, you're more likely to die or have reduced reproduction from malaria, which is not advantageous. And if you are homozygous recessive, you're more likely to die or have reproduced reproduction from sickle cell anemia. So the heterozygote here is going to carry, heterozygote carriers are going to have a survival and reproductive advantage. So the sickle cell allele becomes common in the population because there is an advantage to those heterozygotes. So this is referred to as heterozygote advantage. And if you look at this key, the frequencies of the sickle cell allele, the lighter the color means the lower the frequency of the sickle cell allele, and the more red the color, the higher the frequency of sickle cell allele. And the distribution of malaria caused by this parasite is represented by the dot. So you can see the more dots that you have in a particular area, the more likely or the higher frequency we kind of see of that sickle cell allele. So a really interesting example of heterozygote advantage there. And so as always, you want to make sure that you write down any questions that you have so you can bring those to class. And finally, you want to make sure that you can be able to do a couple of things with your notes. So you do need to explain how variation is produced and maintained in a population. The vocabulary here is going to be really important. So you needed to be able to define all of your new terms used in this presentation in your own words and be able to give descriptive examples. Explain how each source of evolution in population affects variation and selection, and use the Hardy-Weinberg theorem with facility, meaning be able to move through all terms of both equations. So apply the theorem to actual populations is necessary for you to do as well. Those terms is. Let's take a look at this chart to see if we can figure out why this math makes sense. Okay, so on the bottom here, we have the frequency of the dominant allele, that's P, and the frequency of the recessive allele, that's represented by Q. And we can see that if the frequency of the dominant allele, if that's at 0, that means that the frequency of Q must be at 1. And so looking here, Q squared, again, is represented by this